Transcription is the first step of gene expression. In transcription, a sequence of DNA from a template strand is copied onto an mRNA strand. To do this, DNA must first come to RNA polymerase. For this to happen, TATA boxes bind with transcription factors and they guide the DNA strands to the RNA polymerase. RNA polymerase breaks apart the hydrogen bonds of the DNA molecule, separating the two strands. The template strand flows in a 5' carbon to 3' carbon direction. In the RNA polymerase, RNA base pairing rules bond nucleotides to the nucleotides of the template strand and its sequence that you want to copy. The RNA nucleotides are added in a 5' carbon to 3' carbon direction, and as the strand grows, they flow sort of out of the cell because they run out of space inside of the RNA polymerase. Real quick, it is important to mention the base pairing rules of RNA. Guanine with cysteine and adenine with uracil. This process continues until the stop sequence of the DNA is read, in which transcription stops and the DNA is formed back together after it's completely fed through the RNA polymerase. And on the other end of it, you will have a DNA molecule that has been reconnected with DNA ligase to reseal the bonds, and an mRNA strand that has the information you want on it. However, this is not the end of transcription. You still need to edit the mRNA strand. Multiple things happen when you edit the mRNA. First, in the editing of mRNA, you have something called a spliceosome that cuts the mRNA sections. In mRNA, there are sections called exons and introns. You really want the exons because they have sequences that code for amino acids. So you cut those apart from the introns, which do not code for anything. The spliceosome reconnects the extrons and creates a continuous strand. The introns are recycled. The spliceosome is made up of ribozymes and S and RNA. Ribozymes are RNA molecules that act as enzymes.
After translation occurs, a tRNA binds to the mRNA via anticodons, which attach to the codons, which are a sequence of 3 nucleotides. From there, it attaches to a ribosome because it is guided by the tRNA. The ribosome has an E site, P site, and A site. The E site is where it binds, and the DNA and the mRNA is guided from the E site to the P site to the A site in the 5'3" direction. While the tRNA and mRNA are binded to the E site, an amino acid attaches to the tRNA. From there, it then moves to the P site. After it moves to this P site, more tRNA can move into the E site, which then more amino acids can bind to. In the P site, the tRNA releases the amino acid and moves to the A site. When it reaches the A site, it releases the tRNA, and the new tRNA that is in the P site releases its amino acids. These amino acids then bond to create a polypeptide chain, which is connected by peptide bonds. After this chain reaches a certain length, a stop codon, which is in the tRNA, creates a release factor to stop coding for the polypeptide chain. This is the end of the process of translation.